On a warm June evening in the Great Smoky Mountains of Tennessee, something happens that draws thousands of people from across the country to stand in the dark and watch. For 2 weeks every summer, at a precise moment after sunset, the forest fills with light. Not one light, not dozens, thousands of them, blinking in perfect synchrony, a single pulse moving through the trees like a wave. Silence between flashes, then the whole forest lights up again. The fireflies doing this are all males. They are all the same species, and they are all saying the same thing to every female watching from the grass below. We call it bioluminescence. We call the insects fireflies, or lightning bugs, and we treat them as one of the gentler mysteries of summer. The light in the jar, the glow in the field, the thing children chase at dusk that you cannot quite catch and cannot quite explain, but the explanation, when the DNA finally delivered it, turned out to be one of the strangest origin stories in the insect world, because the firefly's light is not what you think it is. It did not begin as a signal. The creature that first made it was not trying to attract a mate. It was not even a firefly as we would recognize one. And the genetic machinery that produces one of the most beautiful sights in the natural world turns out to have been assembled from parts so ancient, so repurposed, so deeply surprising, that researchers who spent their careers studying this insect had to revise everything when the genomes finally came in. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The textbook version of the firefly was, like most textbook versions, a clean story with clean borders. Fireflies, it said, are beetles, members of the order Coleoptera, the largest order of insects on Earth, which contains roughly 400,000 known species. Within the beetles, fireflies belong to the family Lampieridae, and this family is distinguished by one thing above all others. Its members produce light through a chemical reaction involving a compound called luciferin, an enzyme called luciferase, and oxygen. This reaction produces what scientists call cold light, meaning almost no heat is wasted, making the firefly's lantern one of the most energy-efficient light sources ever discovered. The textbook then told the evolutionary story like this. Bioluminescence evolved once in the firefly lineage, in an ancestor of all modern Lampieridae, and from that single origin the light diversified into the many species-specific flash patterns we see today. Different species blink at different rates, different durations, different colors. Male signal, females respond from the ground, mating happens. Clean, logical, elegant. The story held for most of the 20th century because it was impossible to test rigorously. You cannot tell from a fossil whether an ancient beetle glowed. You cannot find preserved luciferin in stone. The fossil record of fireflies is thin, and what exists tells you about body shape, not biochemistry. Then the genomes became readable, and almost everything the textbook said about the origin of firefly light turned out to require fundamental revision. The first disruption came from a question nobody had thought to ask carefully enough. Where did luciferase come from? Luciferase, the enzyme that catalyzes the light-producing reaction, does not exist only in fireflies. It is one of the most studied enzymes in molecular biology, and for a long time its firefly origin was treated as a unique evolutionary invention, something the lineage had built from scratch. The assumption was that luciferase was a novelty, an enzyme that had no prior purpose and no prior relatives. The genomic data destroyed that assumption completely when researchers sequenced the full genome of Equificans photinus, the North American firefly most commonly studied in laboratories, and compared its enzyme genes against the genomes of other beetles, including many that do not glow at all. They found something that stopped them. Luciferase is not a novel enzyme. It is a modified fatty acyl-CoA synthetase. In simpler terms, it is a repurposed version of an enzyme that most beetles, and in fact most insects, already carry for the purpose of activating fatty acids during normal cellular metabolism. The firefly did not invent a light-producing enzyme from nothing. It took an existing metabolic enzyme, one doing a completely different job in the cell, and modified it, through a series of mutations across evolutionary time, until it could produce light as a side product of a related chemical reaction. This single finding reframed the entire question. The question was no longer, how did fireflies evolve bioluminescence? The question became, why did modifying a fatty acid enzyme to produce light get selected for? What was the original advantage? Because the first tiny modification of that metabolic enzyme would have produced only the faintest, most imperceptible glow. Not a signal, not a flash, just a faint chemical byproduct visible to nothing and no one. And yet evolution kept pushing it in that direction. Something about producing light was advantageous long before that light was bright enough or structured enough to serve as a mating signal. The answer came from a second genomic discovery, and it is the one that most completely overturned the old story. Firefly larvae glow. This is not widely known. The glowing adult that flashes through the summer evening is only the final stage of an animal that spends most of its life underground or in leaf litter as a larva. And in that larval stage, across nearly all firefly species, the animal is already luminescent. The larvae glow continuously, not in flashes, and they glow from the same organ in the same position on the abdomen that the adult will later use for mating signals. The larva does not use its glow to find mates. It cannot. It is a larva. It uses its glow, the genomic and behavioral data suggests, as a warning. Firefly larvae are toxic. They contain defensive compounds called lucibuphagins, bitter steroidal chemicals that make them highly unpalatable to predators. Frogs, lizards, birds that eat a glowing firefly larva learn very quickly not to do it again. The glow, aposematic, the biological term for warning coloration, the same principle at work in the bright orange of a poison dart frog or the black and yellow stripes of a wasp. The message is not come here. The message is I will make you sick. The genomic evidence confirmed this by showing that the genes for producing lucibuphagins and the genes for producing light are both ancient within the firefly lineage, present in larvae and adults alike, and that the larval warning function appears to predate the adult signaling function by a significant margin in evolutionary time. The light came first as a defense. The mating signal came second, as evolution discovered that an already glowing animal could repurpose its glow for communication. This sequence, defense first, signal second, is precisely what the gene expression data shows when researchers compare larval and adult tissue. The light organ in the larva is running a warning system. The light organ in the adult is running a communication system. The same hardware, completely different software, and the hardware came first. The third revelation came from the species count, and it forced another uncomfortable rethinking. When researchers used the genomic data to build the most comprehensive phylogenetic tree of Lampreyridae ever assembled, they discovered that the diversity of fireflies was far greater and far older than the fossil record had suggested. The family appears to have originated in Asia, not in the Americas or in Europe as some researchers had assumed based on the richness of North American firefly fauna. The oldest lineages, the genomically deepest branches of the firefly family tree, are Asian. And the age of the family was pushed significantly earlier than the fossils had allowed. The molecular clock analysis suggested an origin in the early Cretaceous, somewhere between 100 and 140 million years ago, in a world of dinosaurs, ancient flowering plants, and in Asia that looked nothing like the Asia of today. The firefly was older than anyone had realized by tens of millions of years, but the number that stunned researchers most was not the family age. It was the rate of new species discovery, because when genetic tools were applied to firefly populations that had been considered single species under traditional morphological identification, population after population split apart. Insects that looked identical under the microscope turned out to be genetically distinct species that had been producing different flash patterns, attracting different mates, and breeding in isolation for hundreds of thousands of years. right under the noses of entomologists who had never noticed because the physical bodies were so similar. The estimated total number of firefly species on Earth approximately doubled in the space of a decade of genomic study. The world was not getting more fireflies. Researchers were simply learning to see the ones that had always been there. The fourth revelation is the one that may matter most for the world outside the laboratory. Fireflies are disappearing. Not slowly, not subtly, dramatically, measurably, in the lifetime of people alive today. A global survey of firefly conservation published in the journal Bioscience in 2020, led by Avalon Owens and Sarah Lewis at Tufts University along with an international team, found that the majority of firefly species worldwide face significant threats. The threats are three, and they interact. Habitat loss removes the wetlands, forests, and meadows where fireflies breed and where their larvae spend years underground. Light pollution disrupts the mating signals that are the entire point of the adult firefly's brief life. And pesticide use, particularly the widespread application of insecticides targeting soil insects, kills the larvae before they ever reach adulthood. The light pollution problem is the one the genomic story makes most bitterly ironic. Fireflies evolved their light originally as a defense against being eaten. Then they repurposed that light as a communication system so precise that different species have different flash codes, different intervals, different colors, all designed to work against the darkness of a summer night. And now we have filled the darkness with artificial light to such a degree that in large parts of the world where fireflies once bred, the night is never truly dark, the signals cannot be read, and the females in the grass cannot find the males in the air. The light we produce has drowned out the light they evolved, the synchronous fireflies of the Smoky Mountains, the ones whose precise coordination draws thousands of tourists each year, are not just a natural wonder, they are a genomic time capsule. Synchronous flashing, the behavior where thousands of males pulse together in a single wave, evolved only a handful of times in the entire firefly lineage. The Smoky Mountain population belongs to one of those rare lineages. The genomic data shows that the synchrony is controlled by a neural timing mechanism encoded in the genome, a biological clock in the brain that locks each individual's flash to the rhythm of the males around it, producing a self-reinforcing wave of light that sweeps through the forest. Why synchrony evolved at all is still debated. The most supported hypothesis from the genomic and behavioral data is that it is a solution to a signal detection problem. In a forest crowded with multiple species of firefly all flashing at different rates, a female of a given species needs to pick out the flash pattern of her own species from the noise of all the others. A male that flashes in perfect synchrony with other males of his species produces a stronger, clearer, more unambiguous signal against that background. Synchrony is a solution to the problem of being heard in a crowd. And it took hundreds of millions of years to get here. From a metabolic enzyme doing housekeeping chemistry in a beetle cell, to a faint glow in a toxic larva warning a predator away, to the neural architecture that lets a forest full of beetles pulse as one. There is one more layer, the DNA revealed, and it concerns something the firefly does that almost no other animal on Earth does. The female of one genus of North American firefly, Photurus, has learned to mimic the flash responses of females from other firefly genera. When a Photinus male, a different genus, flashes his mating signal overhead, the Photurus female on the grass below answers with a flash that mimics the response pattern of a receptive Photinus female. The male descends. The female eats him. She is not starving. Firefly adults of many species do not need to eat at all during their short adult lives. She is eating him for the lucibuphigins, the toxic defensive compounds that the Photinus male carries in his body. By consuming males of a species with more potent toxins than her own, the female Photurus increases her own chemical defenses, and the genomic data has now confirmed that she passes those acquired compounds to her eggs, giving her larvae a chemical head start before they have even developed their own defensive chemistry. The firefly's light began as a chemical warning. The chemical warning became a mating signal. The mating signal became a trap, and the trap feeds the next generation's defenses. This is what 100 million years of evolution looks like when you read it in the genome. Not a clean line from origin to purpose. A tangled, repurposed, weaponized, co-opted series of accidents and selections, each one building on the last, until what you have is a beetle that can light up a forest, trick a rival species into its jaws, and protect its children with stolen chemistry. The firefly in your summer field is not simple. It never was. We just could not read the full story until the DNA let us. On that warm June evening in the Smoky Mountains, when the forest lights up in a synchronous pulse, and then goes dark, and then lights up again, you are watching the end product of a story that began in the body of a beetle that could barely glow, warning something in a dark Cretaceous forest that it would taste terrible. 100 million years of refinement. One pulse of light in the dark. We do not know what that first faint glow looked like. We do not know what first failed to eat the larva that wore it. We only know that something in that darkness decided the glow was worth keeping. And here, in the June dark, the keeping continues. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.